Jennifer Dionne is an associate professor of materials science and engineering at Stanford. Her research develops new optical materials and microscopies to observe chemical and biological processes as they unfold with nanometer scale resolution. She then uses these observations to help improve energy-relevant processes and medical diagnostics and therapeutics. Her work has been recognized with a Moore Inventor Fellowship, the Materials Research Society Young Investigator Award, Adolph Lohm Medal, Sloan Foundation Fellowship, and the Presidential Early Career Award for Scientists and Engineers. And she was recently featured on Oprah's list of 50 things that will make you say, wow. Jennifer Dionne is going to be talking about monsters made of light. So please welcome Jennifer. Thank you.
All right. Great. Cool. Well, thanks so much for the introduction. Can everyone hear me in the back? Okay, people usually complain that I'm too loud, but in case you can't hear me, raise your hand. A huge thanks also to Piero for putting this festival together. It's an honor to be speaking here, and it was great to see all of the art exhibits. I'm really looking forward to tomorrow so I can check everything out more carefully. So I had some great speakers who preceded me. This will be a little bit of a heftier dive for an 8 p.m. on a Friday evening into some science, but hopefully I can convince you that for reinventing the next Frankenstein, light might be the next handy reagent for a lot of chemical transformations. So I wanted to start off and focus on this cool picture I found outside my apartment. I think the rainy season in California has brought with it an abundance of snails, two of whom I found mating outside my home. And the snails are a beautiful example of chirality, a word that derives from the Greek for handedness. So each snail shell has a particular twist associated with it, in this case, a right-handed spiral, that intriguingly propagates all the way down through to the internal organs of the snail. And this means that a right-handed snail can literally only be a good fit for another right-handed snail. It can't hook up with another left-handed snail. They just won't match up. So like the snail shell, it turns out there are many objects in nature that have a certain handedness or chirality associated with it. And those include, for example, DNA, which is always a right-handed spiral, or sugars, which are always also right-handed, or amino acids, the building blocks of proteins, which are always exclusively left-handed. And this homochirality of nature is perhaps one of life's greatest mysteries, but one which we are not quite as good at in reproducing in the lab. So generally, when we as chemists or scientists go into the laboratory and try to make molecules, we wind up getting mixtures kind of like the snail shells. You'll find snail colonies of either left-handed snails or right-handed snails. And generally in the lab, when we're synthesizing molecules, we don't wind up with the homochirality of nature. We instead wind up with what's called racemic mixtures, or 50-50 mixtures. So here's one example of a molecule limonene. Basically, this hexagonal ring that you're seeing corresponds to different carbon atoms that are all arranged in a ring. And you might imagine that if you were synthesizing this molecule, let's say the last atom you need to add on is this hydrogen, it can go to the right or to the left of the ring with essentially equal probability. And depending upon which side of the ring it goes to, you wind up with a molecule that has quite different function. So in this case, the function is actually the smell. So if the hydrogen goes to the right-hand side, the molecule smells like turpentine, whereas if it goes to the left-hand side, the molecule smells like oranges. And the importance of chirality, especially in pharmaceuticals, was brought to prominent attention in the late 1960s when in West Germany, a drug thalidomide was marketed to treat morning nausea in pregnant women. Unfortunately, the non-superimposable mirror image, the molecule where basically one ring went to the other side, actually induced birth defects and in about half of all cases, fetal death. So since that time, the FDA has put a lot of attention into testing chiral drugs, namely drugs that have a certain handedness to it, in a very systematic fashion. And if you look to the market, roughly about 50% of drugs and also 30% of agrochemicals are chiral, meaning they have a certain handedness associated with them. But of those, more than 90% are sold as mixtures because it's so hard to separate those enantiomers, so those right-handed and those left-handed molecules. And those would include the molecule Advil. I'm imagining at least some of you have taken Advil before. And how long has it taken for pain relief? An hour. Okay, yeah, maybe hour, half an hour. That's because only one of the handednesses of the molecule is actually effective in treating pain. The other handed molecule kind of gets in the way. So all of our cellular receptors are essentially proteins, which I said are composed of amino acids. So you could imagine Advil as a drug is supposed to bind with some of our cellular receptors that, say, would reduce the amount of pain we're in. So the right-handed enantiomer can bond properly, but then the wrong-handed enantiomer gets in the way and basically reduces the effectiveness of Advil. So it's sold as a mixture, which means it takes about 30 minutes to an hour for pain relief. If it were pure, i.e. we only had the right-handed molecule, it would take about 10 minutes for pain relief. Also, the antidepressant Prozac is sold as a mixture of both handed molecules, where the wrong-handed enantiomer can cause a lot of the detrimental side effects or the negative side effects like heart palpitations or headaches. And then also, if you look to a lot of agrochemicals like herbicides and pesticides, it's known that the wrong-handed enantiomer can actually leave residues in soil, leading to colony collapse in bees and also organ failure in birds, fish, and larger mammals. So the reason why many of these pharmaceuticals and agrochemicals are sold as mixtures is because separating chiral molecules using conventional chemical methods is really challenging. They're identical in what I'd call all scalar physical properties. Scalar meaning that if you look at their mass, they're essentially the same because you have all of the same atomic constituents. Or if you look at their charge, the charge is also the same. So you can't separate them, for example, using centrifugation, where you would rely on basically differences in mass or weight. You also can't separate them based on charge, say using an electric field. So in this talk, I want to share with you how we can use a new reagent, namely light, and I said I wanted to focus on handy reagents to help separate enantiomers. So light is, of course, an abundant resource, and one where we have essentially photons that are comprised of oscillating electric fields and magnetic fields in space and in time. And light can be polarized. Probably you've worn polarized sunglasses before. And you can also get it to be polarized not just in a linear fashion, but also in a circular fashion. So you can get the electric and magnetic fields to map out a helix in space and time, so that way light is also going to be a chiral object. So we can shape light to be chiral, but unfortunately at visible wavelengths, which have sufficient low enough energy to not, say, destroy or destructively interfere with a molecule, the wavelength is quite a bit larger than, say, a molecule. So the molecules we're interested in separating have sizes on the order of a nanometer. The wavelength of visible light has a wavelength on the order of 400 to 700 nanometers. So to increase the interaction of light with a chiral molecule, or in this case labeled as a chiral nanospecimen, we have to come up with a way of kind of increasing that interaction, and we do so with what's labeled here as a nanocoaxial optical fiber. It's a lot like the coaxial cable that you would plug into the back of your cable television, except it's shrunk down by many orders of magnitude, and instead of carrying electrical signals, it's carrying optical signals. So you can think about this like a very tiny optical fiber. So the circularly polarized light, or that chiral light, enters this nanocoaxial fiber, which has a metallic core, a metallic cladding, and then a very thin dielectric channel. So the light can propagate through the dielectric channel, and this structure essentially acts to shrink down the wavelength of visible frequency light in such a way where it can interact more strongly with the chiral nanospecimen above it. And here's the general scheme of the system that we're putting together. So we have an array of these nanocoaxial fibers. It's basically a very thin membrane that's comprised of a metal where we have these thin dielectric channels. And then we have a mixture of right-handed and left-handed molecules in a liquid above these channels. And when we illuminate with circularly polarized light, basically that light initiates an optical handshake with some of our enantiomers in such a way where you can imagine if we had right circularly polarized light, the right-handed enantiomer would be attracted towards the light, whereas the left-handed enantiomer would stay in solution. And then if this is all liquid, you could imagine that the right-handed enantiomer would simply just go through each of these dielectric or these air or liquid channels to the other side, so we wind up physically separating these enantiomers. So this is a nice complement or an alternate route to many of the conventional chemical routes that would be used. So if you wanted to use a conventional chemical technique, you would have to rely on developing a unique chiral molecule that could preferentially interact with another right-handed molecule. So that's called a chiral substrate. Developing those is a very time-consuming and costly venture, and part of the reason why a lot of pharmaceuticals and agrochemicals are sold as mixtures, because it's just not worth the monetary investment and the time investment to develop a chemical chiral substrate. So the advantage of this approach is you don't have to worry about developing a new substrate. You just come in with circularly polarized light. Okay, so let's see if we can characterize the forces that a chiral nanostructure or a chiral molecule would feel with this chiral light. So first we need to make the coaxial structures. Here we have a thin metallic film, and then we've used a focused ion beam to make a thin dielectric channel in the metallic film, and then we've also patterned grooves around the structure to enhance the amount of light that gets through the aperture. So you can see the scale bar here. The scale bar is about 1 micron, so this structure is many times smaller than, say, the width of a human hair. If we zoom in even further, you can see that dielectric channel in more detail. It's about 50 nanometers across, or about 5 times smaller than most viruses, to give you a sense of scale. Okay, so how would light emerging through this coaxial channel interact with a chiral nanospecimen? Well, what we can do is some calculations that allow us to calculate the pulling force that a structure would feel when it is above the coaxial fiber. So we're illuminating this structure with right circularly polarized light, so the light is mapping out a right-handed spiral. And then we have a right-handed nanostructure that's 5 nanometers in size, and it's positioned above the coaxial structure. So if we calculate the pulling force, you can see that it's quite strong around the dielectric channel when we have right circularly polarized light and a right-handed enantiomer. The pulling force has a magnitude of about 2.5 picoNewtons. So this is an extremely small force. Also, to give you a sense of scale, you can compare this to the amount of force that your fingers would exert on your phone when you're typing a text message. That's about half a Newton. So this is about 11 orders of magnitude smaller. But thankfully, we're dealing with molecules and nanostructures, so that small force is going to be sufficient to pull a molecule through it. And force is a vector. So not only is the force strong along the dielectric channel, but we can also look at the direction with which the nanostructure or molecule would be pushed and pulled. And that direction is going to be towards the coaxial structure. So the structure is going to work a lot like that filter I described, where if we illuminate with right-handed light, that right-handed enantiomer would be pulled towards the coaxial array and eventually pass through it. Now, if we compare this with left circularly polarized light, still having that right-handed enantiomer, which would be similar to, say, having right circularly polarized light and the left-handed enantiomer, you'll see that the pulling force is much, much weaker, and it's actually also repulsive. So if we have the wrong-handed enantiomer, it's going to be pushed away from that membrane. Okay, so how can we quantify these forces experimentally? Well, we do so using an atomic force microscope. So this AFM, or atomic force microscope, is essentially a long cantilever that has a very tiny probe attached to it. The long cantilever is a lot like a diving board. So the diving board is hovering over our coaxial structure, and then we pattern a nanospiral at the end of it that serves as a proxy, so that way we can quantify the amount of forces that are being exerted on the nanospecimen. And like I said, the cantilever is basically like a diving board, so it hovers over our sample. And when we turn the light on, we can see if that diving board or that cantilever gets deflected towards the sample, in which case we have an attractive force, or away from the sample, in which case we have a repulsive force. And the way we know if it's going down or up is we basically just bounce a laser off of it, and we can see if the laser is going up or down on our photodetector. And then we can correlate that with the precise amount of force that's being exerted on the nanospecimen. So like I said, at first, we basically started off with milling a little nanospiral at the end of our atomic force microscope tip, so that way we could just quantify the forces. Here's a scanning electron micrograph image of that spiral, so you can see in 3 dimensions the right-handed spiral. You can also see the top view of the AFM tip where we have a right-handed spiral. This looks a lot like the snail shells that I opened up with. And what we can do is quantify the amount of force that's being exerted when the light is on and when the light is off. So when the light is off, so when the laser is off, we essentially have no force that the AFM tip is feeling. And then when we turn the light on, here we're on resonance for that coaxial structure, the force jumps up. And what we can do is map the forces over a quadrant of the coaxial structure with extremely high spatial resolution. So here we're mapping it out with approximately 2 nanometer pixels. And you can see the force is much stronger when we have our right-handed tip illuminated with right circularly polarized light than when we do so with left circularly polarized light. Okay, this graph is essentially the crux of this part of my talk, where what we can do is now measure out the forces as a function of the height above the aperture. So we have the height ranging in this case from about 40 nanometers to 100 nanometers. So as long as the molecules are kind of in close vicinity of this coaxial structure, we can map out the forces. And we see that for right circularly polarized light, when we have that right-handed nanostructure, the force is attractive, i.e. it's negative, whereas when we have left circularly polarized light and that right-handed structure, the force is repulsive, so the tip gets deflected away from the sample. And this agrees well with our calculated results. So what we're doing now is applying this to small molecule separations. I didn't include the results here. I'm happy to talk about them offline, but we've shown it works now on proteins and DNA. So pretty excited about moving forward. And what I'd like to, I guess, kind of consider this platform as is light initiating an optical handshake. Or if we return to the snails, perhaps an optical smooch that hopefully enables a healthier planet and a healthier population. So I know I have about 8 more minutes and I want to switch gears. I'm going to an even smaller scale and talking a little bit about bacteria. So in this case, I have a picture of E. coli. And while bacteria and bacterial infections may not often make headlines, it's undoubtable that they have a global impact. So in the United States alone, bacterial bloodstream infections cause more deaths than breast cancer, colon cancer, prostate cancer, and HIV combined. And in developing countries, about 30% of all deaths are due to bacterial infection. And if you look globally, about 1 child dies every 20 seconds from a bacterial-related illness. So such dire statistics are due in part to the way in which bacterial infections are often diagnosed. So if you were to go to Stanford Hospital and they suspected that you had a bacterial infection, what they would do is they would generally collect a blood sample from you. And in that blood sample, which roughly is going to be about 5 milliliters, maybe up to about 20 milliliters, you're only going to have 1 bacterial cell per milliliter. So our bodies are an incredible sensor of bacterial infections, and yet it's very hard for doctors to be able to diagnose bacterial infections because it's hard to find that needle in a haystack. Essentially, there's only 1 bacteria per milliliter of blood. So what they'll do is they'll culture the cells. So they'll essentially wait for bacteria to grow and multiply until there are a sufficient number of bacteria where they can detect it. And then what they'll do once they've cultured it is they'll screen for antibiotic susceptibility. So they see, is a bacteria present? Once they've cultured enough bacteria, then they'll usually use mass spectroscopy to be able to figure out which bacteria it is. And then what they'll do is they'll take their plate of bacteria and put antibiotic patches on top of the bacteria and figure out which antibiotics the bacteria is responding to. So this is kind of the gold standard for bacterial infections. And depending upon the type of bacteria you have, it can take anywhere from about 1 day up to 40 days. So 40 days is pretty typical for slower growing bacteria like bacterial tuberculosis. So meanwhile, you're in the hospital and you're not feeling great. And according to the CDC, about 50% of patients in that time will be treated with antibiotics based on empirical guidelines that the doctors are using. But of those, you're treated usually with the wrong antibiotics or with the wrong dose of antibiotics, or you're unnecessarily treated because maybe it wasn't a bacterial infection after all. So this is kind of a treatment procedure that's giving rise to a lot of antimicrobial resistance, which I think undoubtedly in the next few decades might become one of the bigger problems that we're faced with. Okay, so my team at Stanford is working on a new approach, again, using light as a handy reagent to help diagnose and identify bacterial infections, hopefully in a much more rapid fashion. So the idea is that you print a patient's blood essentially onto a postcard where each pixel of that postcard corresponds to a single cellular droplet. And then you take a picture of that postcard. So you have a postcard, each pixel is a single cell. There's a laser that illuminates that postcard, and then based on how each pixel scatters light, we can detect from the color of the pixel what type of cell it is, whether it's a red blood cell, a white blood cell, or a bacterial cell. So the particular color comes from a process called Raman scattering. Basically, the laser is deflecting off of the cells, and based on the amount of energy either lost or gained by the laser light, i.e. their color, and some machine learning algorithms, we can identify exactly what type of cell it is. So in the next couple of minutes, I just want to talk you through how it works. I'll mention that kind of the advantage of this approach is that it requires no sample-specific preparation. You could simply imagine just printing out the droplets of blood onto a postcard, and it doesn't rely on any cell tagging, so there's no specificity in terms of the sorts of bacteria that we could detect. It should be quite generalizable. Okay, so in kind of the first rendition of this diagnostic, we're printing these single cellular droplets essentially using microfluidics. So here's just a video of droplets of single cells of either blood or bacterial cells being printed using microfluidics onto an array. And each of these droplets contains not only a single cell, but also these metallic nanoparticles that can enhance the amount of light we see. So it can basically enhance the color contrast that we're seeing. Okay, and then like I said, what we do is we essentially take a spectra. So we look at the color of each of the cells. And if you look at basically the Raman spectra, or the color of various bacteria, if we integrate for a long time on each pixel, then we get a pretty clean spectra, and we can pretty clearly distinguish between the different types of bacterial cells. So far we've looked at the 31 species and strains that are most commonly seen at Stanford Hospital. But since we're processing, say, 5 milliliters of blood, and there are billions of cells within there, we can't integrate on each pixel for even seconds. We want to get to the sub-second and ideally sub-microsecond level. So here's kind of a typical example spectra if we have single cell data and just a 1-second acquisition. And you'll notice that across these different bacteria, the signal is quite noisy. In fact, by eye, you can barely see any difference between different bacterial species and strains. The only spectra that looks distinguishable is, for example, this blood cell. So we can easily tell blood cells from bacterial cells, but you can't really tell bacterial cells apart. So this is visualized a bit more quantitatively here. There's something called principal component analysis, where you can kind of figure out the features of different colors or different spectra. And you'll see that the blood is easily distinguished from bacteria. But if you kind of zoom into this bacterial blob and you do another principal component analysis, here on the 31 bacterial species and strains, they're all kind of overlap. So you can probably distinguish about 7 different colors. You definitely can't distinguish the 31 different colors and where they're lying here. So what we've done is worked with our collaborators in computer science to develop a convolutional neural network that allows basically the computer or the AI to more clearly see the differences in the color of these cells. So we collect 186,000 spectra. We have 31 strains, 3 integration times, basically 2,000 spectra per condition. So we have 2,000, say, single cell data. We take 1,600 of that and use that as training data, leaving out 400 that we'll use as test spectra on our neural network to see how well it's performing. And then we have a slightly different experiment where we have the same spectra, 2,000, but we remove, again, 400 that we use as our test spectra, 1,600 that we use as our training data. So we do that basically 4 times, but across the 2,000 spectra collected across 8 different conditions on 31 strains and for 3 integration times. So here's just kind of a roadmap for how the convolutional neural network works. Pretty amazing that we've trained, say, a Google search to be able to identify what the content of images are. You can think about our spectra as essentially being a simplified image. So at the end of the day, images are just pixels, where each pixel corresponds to a certain number corresponding to the intensity of a certain color. And here we essentially have essentially the same thing as an image, but here just laid out along one dimension. Okay, so when we use our neural network to be able to identify data that looks even noisier than this, we can figure out the accuracy with which we can identify single-cell noisy data. So here are those 31 bacterial species and strains. The different colors correspond to different antibiotic treatments that you would use on each bacteria. And you'll see that generally we're doing quite well in terms of our predictive accuracy. So in some cases, for example, on some of the staph strains, we have accuracies that are above 97% and 100%. So even though by eye, to me, the spectra looks just like noise when we're integrating below 1 second, the AI can clearly distinguish all of these bacterial species and strains. The ones it struggles the most on are on MRSA, but it can't tell the difference between a clinical MRSA and basically an isogenic MRSA, where basically physicians have taken out the strain that they know causes antibiotic resistance and removed it. So for all practical purpose, you'd never actually see this in a clinical setting. You would basically just see MRSA versus MSSA, the antibiotic susceptible strain, and there we have classification accuracies that are approaching 90. Okay, so if we group things according to antibiotics that a doctor would use to treat each of these bacterial infections with, you'll notice that essentially all of the classification accuracies are above 80. If we remove those isogenic strains of MRSA, then all of our classification accuracies are above 90. Okay, so that brings me to my conclusions. Again, like I said, sorry for the kind of heavy science following an amazing presentation on San Francisco counterculture. But hopefully I've shown you that light can be the next handy reagent for kind of reinventing Frankenstein. First part of my talk focused on chiral sorting, how we can use these nanocoaxial structures illuminated by circularly polarized light to selectively trap and then sort chiral nanostructures based on their handedness. Then we talked about bacterial identification and how using a process called Raman scattering, or essentially the color of a bacteria combined with machine learning, to basically rapidly and sensitively identify bacteria and their antibiotic susceptibility. And then if we're thinking forward about, say, Frankenstein at 300, I think it's interesting to think about whether we could use light, for example, to dynamically monitor drug-cell interaction. So in real time, we could see how a drug is interacting with a particular cell and hopefully inform the next generation design of drugs. Or perhaps more intriguingly, using some of the insight from this chiral sorting, using light to control ion channels without cell-specific tagging. So some of you may have heard about optogenetics, using light to control action potential spiking. It turns out a lot of the proteins in our neurons are also chiral, and this technique may open the way of using light without having to introduce different genetically encoded fluorophores to enable selective action potentials in synaptic transmission. So with that, a huge thanks to all of the funders of our work, also to my lab, and all of you for your attention. I'm happy to take any questions.
In the black here?
How would you describe the attractive force? Like, how is that being attractive for the chiral scattering?
Yeah, so you're asking me how I might describe the attractive force between, say, right circularly polarized light and the right-handed enantiomer. So the heuristic way of thinking about the attraction is just kind of considering maybe the attraction or the selective binding that you would have, for example, with a screw being threaded the right way into a wall. So if you're threading it the right way, it kind of clicks in there right away. If you're screwing it the wrong way, it doesn't have good binding. The more scientific way of viewing it is basically these chiral molecules are miniature electric dipoles and magnetic dipoles. And we think about electric dipoles as just being linear charges, like a positive charge and a negative charge that are aligned with each other. So that's your electric dipole. A magnetic dipole, I guess you can think about it as coming from a current loop, right? So you have a magnetic field whenever you have current going in a loop. So one of the simplest chiral structures is a spiral. And you can think about that electric dipole and that magnetic dipole. If the current is going in one direction, you get a spiral in one direction when you add those up. And if the current is going in the other direction, you get a spiral going in the other direction. So when you solve Maxwell's equations for electric dipoles and magnetic dipoles interacting with circularly polarized light, you wind up finding that there is a preferential force, and depending upon which way the magnetic dipole is pointing, that force is either positive or negative. So the attractive force basically comes from the magnetic dipole being aligned with the magnetic field of light in the right way to get an attractive force.
Maybe you'll be around for a couple minutes if people still
Yeah,
have
or
questions. Oh, okay. One more quick question.
Sorry. It's very interesting that you use light polarization to separate the chiral compound. But have you thought about that, how to scale up to the industrial scale? And have you ever tried in the laboratory, for example, use Advil as an example, have you tried that to separate a large quantity and to compare the cost with the conventional way?
Yeah. So we can definitely talk a little bit more over the art. So right now we have mostly focused on kind of smaller lab scale prototypes where we have microfluidic chambers that are working on approximately up to about a gram of material. In terms of scaling it up to an industrial level, I'd love to kind of talk with the broader audience and see how that might be possible. I think the first thing we need to focus on with cost is making sure it can work, say, without laser illumination and with a light-emitting diode. And then you could imagine using a, say, stamping approach to make this array over a larger area, and then illuminating, say, a conventional chiral compound just with a light-emitting diode. So that way, instead of having a particular chemical chiral structure, you could just have this coaxial array illuminated by a light-emitting diode and then have the appropriate physical separation of chiral structures.
Okay, thank you so
Yeah.
much.